First, I would like to thank the organizers for giving me the opportunity to present our work. And secondly, I would like to thank Professor Bouchard for introducing my topic, even highlighting the gene I'm going to talk about. So I'm going to talk about much simpler stuff, just mutation in a single gene that will cause obesity. All these, so many different factors can cause obesity. Now, if we can find some extreme cases where mutation in a single gene, a single nucleotide change can cause the child to be really have insatiable appetite from very early on. So I'm going to talk about primarily the MC4 receptor. Towards the end, I will talk a little bit about the MC3 receptor. So we know that obesity is much more than meets the eyes or the need for us to change the road signs, right? Otherwise, we wouldn't be meeting here. So we've been working on this system, the melanocortin system. It consists of 4 hormones, primarily of 4 hormones, alpha, beta, and gamma, melanocyte-stimulating hormone, as well as ACTH. All these 4 hormones are derived from the post-translational processing of a single prohormone, POMC, pro-opioid melanocortin. So in our anterior pituitary gland, we have the first enzyme, PC1. It will cleave POMC into ACTH that will regulate your adrenal proliferation as well as steroid production. In the hypothalamus, as well as in our skin, we have another enzyme called PC2. This will further process these peptides into alpha and beta and gamma MSH. So these 4 hormones have to act through 5 receptors. So these 5 receptors, number 1 to 5, that's just based on the sequence of the cloning, will mediate the 4 hormones action. The first 2 are the classical receptors we've known for many, many years. The first one is our classical MSH receptor that will regulate our skin pigmentation. The number 2, ACTH receptor, regulate our adrenal gland function. Both of these receptors were first cloned by Dr. Roger Kong, who is in the audience. And then we cloned several additional receptors, 3 to 5. So number 3 and number 4 are primarily expressed in the brain. So they regulate different aspects of energy homeostasis, whereas number 5, again from Dr. Kong's work, is involved in regulating sebaceous gland secretion. This melanocortin system is very, very unique in that we have 2 endogenous antagonists. In all the other G protein coupled receptors, we never know of any endogenous antagonist, but in our system, we have 2. The agouti will antagonize the MC1 receptor, so both the balance between agouti and the MSH will determine how much skin pigmentation you have. And the agouti-related protein will antagonize the 2 neuromelanocortin receptors, MC3 and 4. So again, the final function of these 2 receptors are determined by the balance of the agonist, the MSH, as well as the antagonist. And then we have additional ancillary proteins that can further modulate this receptor's function. So these receptors are downstream of leptin. Our adipocyte produce leptin that can cross the blood-brain barrier, binds these receptors in the arcuate nucleus. We have 2 subsets of neurons there. One expresses POMC, the precursor of our hormone, as well as another anorexic peptide, CART. And then the other one expresses MPY as well as agouti-related protein, AGRP. So from POMC, you have produced alpha message that will stimulate the 2 receptors, and the AGRP will inhibit the 2 receptors. I highlighted a few molecules on this slide in red. When you have mutation in any single gene of these red molecules, encoding these red molecules, you will have monogenic obesity. So these kids will be very, very obese when they are really young. So this, again, is biology controls of environment or later behavior factors. So this one just to highlight where these two subsets of neurons are located in the arcuate nucleus. So this is the third ventricle, right? And then the green neurons are the AGIP antagonist neurons. They are located more centrally, whereas the POMC neurons are located more laterally. So this slide summarize from Michael Schwartz, summarize what these two receptors do. Again, your adipocyte produce leptin, activate our two receptors. When you activate the MC4 receptor, you will change two aspects of energy balance. You will decrease food intake and increase energy expenditure. So both of these results in a negative energy balance, right? Again, food intake contributes about 60% of the phenotype, and then the 40% was contributed by energy expenditure. The MC3 receptor is really much less well understood. Again, Dr. Kong contributed a lot of the original research into this receptor. I'll talk a little bit more towards the end. So when the MC4 receptor was first cloned, we didn't know what function it was doing. We knew that it was expressed in the brain. So when Dennis Hassan, together with Roger Kong, knock out this gene in mice, it was shown that indeed you have dramatic increase in food intake, similar to like the naturally occurring agouti yellow mice. When you follow the body weight of these knockout mice, so upper there is the knockout, where male, females, right? And down below, the black lines are the wild type. So you can see that they have a dramatic obesity, body weight almost twice as much as wild type littermate. Those red lines were heterozygous mice. So you can see that when you have just one normal copy, you cannot maintain a normal energy homeostasis. Your body weight is higher than the wild type littermate. So that paper was first published in 1997. Very quickly, two groups, Stephen O'Reilly and at Cambridge, Philip Vogel at the Pasteur Institute, reported the first MC4 receptor mutations in 1998 in Nature Genetics. Since then, we have more than 150 mutations reported. So this is just highlighting some of the missense mutations. I didn't include any of the truncation or frame shift mutations. You can see that in a very small molecule, 330 some amino acids, we have more than a third or 40% of the amino acids that has been found to be naturally mutated in humans. So when I first started working on this, I asked two very simple questions. First, do these mutations indeed cause a loss function in the protein? Secondly, what are the molecular defects? So again, these receptors are G protein coupled receptors. So when the receptor becomes active, they will generate second messenger for this is the cyclic AMP. So we measured, okay, how much cyclic AMP you can produce after stimulating with different dosage of the agonist. Here we use a super potent analog of alpha message. You can see that a wild type has a nice dose response curve, right? Some mutant receptors indeed can respond. Although they were identified from obese patients, they could respond. But you can see that those mutations highlighted in red, they have dramatic defect. Either they're totally not responding or they respond really, really badly compared with the normal receptor. So we next ask, is that defect in signaling due to a defect in binding to the hormone? So this is a hormone binding assay. We say how much binding we can detect. These receptors are located on the cell membrane. So we label the peptide hormone and then incubate the cells. you can see some of the mutations has binding similar like a wild type, but again, those red mutant receptors have defects in ligand binding. So this could potentially because the receptor is defective in recognizing the hormone, or it can also be because the mutant receptor simply cannot get to its normal location, the plasma membrane. So when extend these cells expressing these mutant receptors, we show that, okay, the wild type receptor in the non-permeabilized situation where the antibody can recognize the extracellular epitope, the wild type receptor is expressed nicely on the cell surface, right? Some of the mutants are also expressed nicely on the surface. But again, those red mutant receptors, you cannot see much staining, right? But now if we permeabilize the cells, we can see that indeed the mutant receptors are expressed, it's just they are not transported onto the cell surface. So again, the major conclusion is that this obesity indeed is because the protein has a trafficking defect. Mutation causes misfolding, so the protein cannot travel to the cell surface. Earlier I mentioned that when you have mutation in leptin receptor, or POMC, or the enzyme PC1, right? You can all have monogenic obesity, but of the numerous genetic studies, we found very few patients have those mutations. Whereas in this receptor gene, we found a much higher percentage. In some of the Caucasian population, up to 6% of early onset severe obese kids have a mutation in this gene. So that's very, very high. So recently we showed that, okay, when you have defect in MAP kinase signaling, this is another signaling pathway that this receptor use, can also potentially contribute to obesity because we found some mutations that have no defect at all. So whether they are indeed causing obesity, we were not sure. But now we show that, okay, maybe they are causing obesity by some other mechanism in not just the classical second messenger cyclic AMP. So we classify these mutations into 5 different classes. Some of the frame shift mutations can be class 1, where you don't have normal protein production. And the class 2 is the most common one. The mutant receptors are retained inside the cell, so they cannot get onto the cell surface, so therefore they are not functional. And then we also have identified mutations that cause defect in binding or in signaling. Again, some of the mutant receptors have no defect. So now you found this patient has this mutation, right? You know what's the cellular phenotype, how you can let the patient benefit from your work. So potentially you can have 3 strategies to have personalized medicine to treat. The first speaker mentioned that obesity is not a single disease. Indeed, obesity caused by mutation in this gene is not a single disease, because when you have a different mutation cause a different defect, you have to treat them differently. The same way like our cystic fibrosis is not a single disease either, right? So potentially we can have 3 strategies to correct these mutations. The first one is for correcting those nonsense mutations where you can use these antibiotics to let the translation go through. The third one is that, okay, if you have a mutation that's not responding to the endogenous agonist, can you synthesize a small molecule or other agonist that may potentially can bind to the mutant receptor and then activate the receptor, right? But we were focusing on the second strategy, what's called pharmacological chaperones. So these are small molecules that can cross the cell membrane, go into the ER, the endoplasmic reticulum, bind to the misfolded receptors, and then help them to achieve the native conformation Once they achieve that native conformation, the cell quality control system say, okay, you're normal, you can get out, you can exit the ER and then transport it under the cell membrane. So they are functional. So I'm going to present some of the work. The first compound was first described by Millennium Pharmaceuticals. The second one was described by a French company, Ipsen. These are, again, a small molecule hydrophobic compounds that specific for our receptor. So if you treat the cells expressing the mutant receptor with these compounds, the first one, the Millennium compound, is a low affinity compound, so the effect is a little bit less dramatic, but if you see the high affinity compound, again, before, after treatment, you see a dramatic change in the cell surface expression of these mutant receptors. Both of these receptors were identified, both of these mutations were identified in Italian patients. So this is just quantification by flow cytometry. We show that indeed you can increase the cell surface expression. Even for the normal receptor, when we don't have any mutation, the cell surface expression can also be increased. So potentially this strategy might be used to treat common obesity. So next we ask, okay, now you try to coax them onto the cell surface. Are they functional? So we stimulate with the agonist. You can see now, before you only have 10% of activity, now you have 80 or 90% of activity. Again, we studied these compounds in a number of different mutations. All these were identified from obese humans. So we can see that the low affinity compound has favorable effect. Some works well, but some not. But the high affinity one, again, can correct all the mutations that we studied. So again, in some of the mutations we see, for example, this P78L, that means your 78 codon position was changed from proline to leucine. Originally, you don't have any activity. After this correction, you can achieve 80% of the normal receptor function. So potentially, we can use these compounds to treat patients with these class 2 mutations. Indeed, we just reported that, so the first two I talk about were antagonists. So in the animal or in humans, you have to wash those antagonists out before the endogenous agonists can work, right? So we were also asking, okay, can we have an agonist that you don't need to wash away that can potentially also act as a pharmacological chaperone? So this one, THIQ, is a small molecule agonist. We can see that indeed this can also act as a pharmacological chaperone for at least some of the mutations. Some didn't work so well. So I'm going to spend just 1 or 2 minutes on the MC3 receptor. This one does not change your food intake. So people were not so excited about it because, right, it doesn't regulate your food intake. But when two groups, including Dr. Roger Kong, knock out this receptor, they found out indeed food intake no change, body weight no change. But now if you analyze the body fat, it was shown that, okay, the knockout mice has twice as much as body fat as the normal wild-type mice. So you can see the fat cells, the size are also larger. In another group, again, this was done at Pennington Biomedical Research Center. They showed that using a different strain of mice, these mice are usually much more leaner. They found out that when you knock out the MC3R, indeed the phenotype is much more dramatic than the more obese prone lamb. So in the late 90s, there were a number of genetic studies indicating that the locus that's encoding the MC3 receptor is associated with obesity and type 2 diabetes in humans. So after that knockout study, people went back to sequence the gene in those patients. Nobody found any real mutation, only 2 SNPs. But in 2002, there was one paper from Singapore reporting that there was a potential mutation. So we studied what is the functional implication of that mutation. We showed that indeed, this I183M mutation that is expressed on the cell surface because it compounds the ligand, right? But it generates no signaling at all. So again, this is a class 4 mutation, right? So right now, in this receptor, in the MC3 receptor, it's still kind of controversial whether indeed this mutation will cause human obesity. But I think there is one problem with a lot of the genetic studies is that they, the same way they did with the MC4 receptor, they only measure the BMI. But we know that at least from mice, the MC3R does not affect BMI. It affects your adiposity. So if they go back to study more, focus more on adiposity, perhaps we can have a better conclusion on this, whether this mutation in the MC3 receptor gene is causing human obesity. But right now, in this, we have much less mutation, but we still found 20-some mutations, and there were a number of them that have dramatic effect on the receptor function. So our long-term goal is to be able to do better than these artists, right? We hope to find personalized drugs that can indeed treat this monogenic obesity. Thank you very much.
Thank you. So that was really interesting. So there are some thoughts about this selective leptin resistance, and I guess it relates to kind of the cardiovascular system, and I know we have some better experts here than me on this, but I guess my question would be, if you agonize these pathways and they may well contribute to the hypertension aspects of obesity, how might your chaperones or your approaches be influenced?
Yeah, that's a great question because, I mean, a number of years ago, almost every major pharmaceutical company has a big program on the MC4 receptor. After Stephen O'Reilly published that paper in New England Journal of Medicine, as well as Professor Hall's work, Alan Mark in Iowa, a number of people showed that indeed, the MC4 receptor activation will cause increased blood pressure. So the mechanism, whether we can dissociate the energy balance with the blood pressure, remains still to be investigated. Potential is that, okay, now there is another very important concept in GPCR therapeutics, is called functional selectivity. So this receptor, I just showed 2 signaling pathways, right? But indeed, these receptors can also signal through a number of other signaling pathways, including affecting calcium or inhibiting the G protein, decreasing the cyclic MP production. So whether we can find a way or dissociate the mechanism regulating energy balance, whether it's primarily through GS or MAP kinase or GQ or GI, versus those regulating blood pressure, is a very important question. I think with functional selective or biased ligands, there is still a potential. We don't know. Yeah.
Dr. Tao, your work very nicely shows that in the hypothalamus there is a system that is exquisitely sensitive, and that even a partial defect of one allele of 2 genes can cause morbid obesity. that raises the question, I don't know if anybody has addressed this, what potential diet gene interactions there are, and what that might tell us eventually about obesity prevention, because is this system sensitive to diet or other environmental factors?
Yeah. I'm sorry, because of the time constraint, I tried to say it in the abstract. I didn't prepare any slides. But indeed, Philip Froger followed patients of 3 generations. The grandparents who were born during the Second World War, right? And then the kids, and then the grandchildren. So they all have the same mutation, but the grandchildren, they have the body weight curves in the 1940s or 30s, right? But if you go back to look at those health records, although the grandparents have the mutation, so predispose them to obesity, they were never overweight, right? And then the parents were kind of overweight, but then the grandchildren are all really, really obese. So yes, indeed, there is a gene, environment, gene, diet interaction. When you don't have enough food to eat, you will not be obese, although you have a strong appetite, strong, yeah. Thank you. Thank you.